after his PhD and postdoctoral tenures, he has been working in the area of biotechnology as well as related areas for a long time. And today he'll be talking to us about specifically neurodegeneration, and within that, the role of certain biomolecules in reversal of stress-induced neurodegeneration. Professor Pravir Kumar, we have students of intake in computational biology here, as well as some of the faculties and other PhD students, etc., who are interested in the broad domain of computational biology. So I welcome you to get started with our computational biology seminar. Thank you.
Thanks for the introduction, and I extend my thanks to IIIT Management and Ms. Chen and you for inviting me for this occasion, for this seminar series. So today I am not going to discuss about the computational biology. To tell you very frankly, I am a physiologist, so I'll tell you physiology as well as some proteomics stuff. So you have to be bear with me, okay? It's not like it's interesting. I think that it's going to be interesting. So start with talk. I hope you all know about the Alzheimer's disease and Parkinson's disease. Alzheimer's is loss of memory. So there are different stages of the Alzheimer's disease. It's starting from temporal portion. This is the temporal area, and initially it goes to the parietal lobe. Okay, and after that, when its severity comes, it's go to the frontal lobe, and finally, if it's very severe, in the occipital lobe. So, if you can see here that right starting from this area, temporal area, migrating to this parietal area, then frontal area, and finally in the occipital area. So these are 4 different stages, which is divided into 7 parts, depending on the severity of Alzheimer's disease. Likewise, Parkinson's disease. If I may tell you that how this is progressing, so it starts with like a stage 1 unilateral, which there is no much problem, people can stand freely, walk freely, no problem. But in the second, it's a bilateral or midline involvement, which means little bit bending towards in the walking direction. Third stage is impairment in the writing reflex. Okay, because if it's a reflex, if you can go with the knee jerk experiment. I don't know how many of you know about the knee jerk experiment. So that reflex is not proper. And fourth stage is where a patient cannot walk without support. And fifth is the completely injured state. So I have told you about the Alzheimer's disease pathophysiology progression and as well as this Parkinson's disease. So now we have a biggest competitor in India, that is Bollywood. So we are going neck to neck. We are doing a study in the Parkinson's, Alzheimer's and different diseases, and our Bollywood is doing extensively work for the neuroscience fraternity. I hope you all have seen these movies, and if you remember this, you see these movies, these students, you, me and Ham and Black, is dealing with the Alzheimer's disease, Karthik calling card schizophrenia, this is amnesia, my name is Khan, is again this higher grade autism, Burfi also autism, Gujaris, it's injured spinal disease. So it's nice that you can have a lot of different kind of exposure from Bollywood also. So, and that is why we are regarding these are our competitors, because what we are thinking, they are translating in the movie. So start with the Alzheimer's disease pathophysiology. I hope you all know about the A beta. A beta is the byproduct of holo APP, and as you can see here, this is the holo APP, and this is the beta secretase and gamma secretase. When it cleaves, this A beta is obtained, and this is amyloid intracellular domain. So if you go in detail about this A beta polypeptide physiology, you will find that this is the main culprit for Alzheimer's disease, Parkinson's disease, and inclusion body myositis. Inclusion body myositis is a muscular disorder, but the hallmark feature of this disorder is that A beta is expression. Like in Parkinson's disease, this is also a neurodegenerative disorder, but neuro as well as muscular disorder, because here muscular motor impairment is occurred, while Alzheimer's disease is dementia. Now, someone can ask you what is the striking difference between Alzheimer's disease and Parkinson's disease. You can say in Alzheimer's the loss of memory, but in Parkinson's loss of motor movement, loss of muscular activity. So this is a striking difference between this. Means here patient's memory is okay, but here patient's memory is not okay. Here motor function is okay, here motor function is impaired. While this is exclusively a disease which showing the muscular atrophy or dystrophy or muscular impairment. So, but in these three diseases, A beta playing an important and major role. So let's talk about some protein chemistry. Yes, please.
One question is like you mentioned about Alzheimer's, Parkinson's, and IBM. So what are the priorities?
What is the
Priorities.
prion disease? Prion disease means, I'm just right now talking about the A beta. Okay? Prion is the aggregation of the protein, but here we are talking, obviously, most of the neuronal disorders is a byproduct of aggregation of protein. You can decipher this. But here, A beta, exclusively, we are discussing about the Alzheimer's, Parkinson's, and IBM.
So, no, I just asked,
Yeah.
might be this slightly away from what actually, because prion is also considered as a neurodegeneration.
Yes, you're right.
And also, some of the mechanisms what you have actually mentioned in the prion, like secretase and gamma secretase. Even in prion also, people have actually scrappy prion, people are actually looking into this same mechanism.
Yes, yes.
So, couple of questions is that why prion is not being put along with the answer? That's because of the A beta you are looking at it. Is it because
Yes,
of it?
exactly. And important thing that if about you can say 60,000 groups across the globe is working on this neurodegenerative disorders. So if we start linking, so most of the neuronal disorders is linked together. Whether it's a HP, whether it's a ALS, whether it's a polyglutamin disease, whether it's a prion disease, whether it's a Alzheimer's. So it's interlinked. But yeah, we have to decipher this. But here exclusively I am talking about the A beta parameter. Okay. So in this room, I hope everybody has the A beta monomer. So A beta monomer is not at all a problematic issue. But the moment it becomes oligomer, it's going to be disturbing for the body physiology. And the moment it becomes a fibril, it's start taking disease progression. So suppose in this window there is a curtain. So it's very hard that a mosquito or any insect can come inside. But what would happen if you could completely open this? It's very easy coming and going out. So between the two synapses, what is happening? Between the two synapses, the neurotransmission occurs. But suppose if you put a seal between the two synapses, what would happen? There will be no more easy neurotransmission. And because of that, what would happen? No further synaptic function. And you know, the body is always regarding this Lamarck's theory. If there is no use, there is no point to stay there. And this is why these two synapses are start degrading and this neurodegeneration phenomenon is occurring. So if somebody will ask you what is the neurodegeneration, you can easily say that okay, when the neurons are not functional, when the neurons have nothing to do, it will start degenerating. But this is not only one mechanism, there are a lot of molecular things behind this, but this is just a layman statement. So let's talk about the protein quality control, and you can see that inside our body, approximately 30% of proteins are discarded because it's not in the proper folding. And as you said, Brian, here comes to this. Because you know, the proteins are not properly folded and it start aggregating inside our body. Not only that, there are different mechanisms like inclusion bodies. Because most of the protein sometimes traps in the inclusion bodies. So what happens? This misfolding is called the protein aggregation. And for that aggregation, what is happening? It requires something to maintain or to give a proper fold of misfolded or aggregated protein to a functional protein. And for this, cell required machinery, which is chaperoning machinery or ubiquitin proteasome system. These two machinery is try to help to maintain the protein homeostasis. How? Chaperone is trying to give a proper fold of misfolded protein, but there is a threshold. Beyond that threshold, chaperones cannot work. And then what happens? Chaperones is saying that, okay, I cannot make this protein any more foldable form, so it has to be discarded. But now the question is here, who will decide that what protein should be discarded and what protein should be kept inside the cell? And from here, there is a there is a thing called E3 ligase. This is the ubiquitin E3 ligase. So what is happening? Suppose, if in the internal cellular milieu, if some proteins are or not functional or protein has to be degraded, what will happen? Ubiquitin proteasome system or UPS machinery is recruiting E1, E2 and E3 ligase to clean off this. So how it happens? E1 is the activator enzyme which is triggering the process, while the E2 is conjugating enzyme which giving some molecule that, okay, take one ubiquitin molecule and bind over there. And after that what is happening, there is a series of molecules which is called the polyubiquitin chain. And that polyubiquitin chain has also some peculiarity that it is only bind in certain residues. And most important residues is K48 and K63 residues of the lysine residue. So what happened? These polyubiquitin chain is bind to a protein which has to be discarded and then it cleaned. Since this is not a continuous process, so it has to be stopped. Otherwise, you know what will happen? It can cleave or it can discard other proteins as well, which is functional for our body. So what has happened? This E3 ligase is exclusively decisive partner or component which can decide, okay, which protein should be kept inside and which protein should be discarded. In order to stop this reaction, there is another enzyme called D-ubiquitin enzyme. What is happening? This dissociation the complex of the E1 into an A3 and the entire process is stopped. Hope I am clear?
What is the role of heat shock protein?
Yeah, heat shock protein is just trying to give you a proper shape of the misfolded or aggregated protein.
So it's a HSP 90 or HSP 70?
It's a series HSP 110, HSP 90, 70, 60, 40, 27, 10.
It's all in the eukaryotes or in the...
In prokaryotes as well.
These are chaperones.
Yes, chaperones. Yeah. So now, come to the heat shock protein. If you can see here, there are different inserts like heat shock, amino acid analogs, oxygen-free radicals, transition of heavy elements, metabolic impairment, and these are the pathophysiological features or states like fever, neurohormonal stress, hypertrophy, oxidation. There are so many things. Oncogenic signal, proto-oncogenic signal, development, differentiation, growth, cell, hypoxia. So there are non-stress conditions as well, pathophysiological condition and environmental condition. So there are many inserts which trigger cells to combat with the adverse conditions. Suppose what would happen if we are going in this summer season of Delhi. We need something to cool off or at least an umbrella. Otherwise we cannot survive. Likewise in our body, if there is any adverse situation, these are the line of defense which can try to combat the adverse condition by upregulating the heat shock proteins or molecular chaperones. So you know molecular chaperones is an English word which tells their daughter is going to a party along with their grandma. So that the grandma can take care of that granddaughter. Okay? So here chaperone is simply assisting body to cope with that adverse condition. So you see, the father of medicine, Hippocrates, what he said, give me the possibility of fever, I will cure the disease. That which drugs fails to cure, the scalpels can cure. That which the scalpels fails to cure, heat can cure. If the heat cannot cure, it must be determined to be incurable. So it was initially that, okay, if something has happened, okay, it has to be if the body temperature is elevated, things can be under control. So that was the power of heat shock protein when it was invented. Now come to some stresses. As I said you, like this hypoxia, and I'll also give you some glimpse of the environmental stress. So what happened, I will show you in my next few slides, two different kind of disease model, animal model. One is under hypoxia and another is under rotenone treated. And I'll show you how the neuronal degeneration occurs and how to reverse the effect of these two stress mediated diseases. So we start with hypoxia. In the hypoxia, what is happening that, and this is the, as you know, this is the angiogenic pathway. I don't know how many of you know about the angiogenic pathway. Angiogenic pathway is just increasing the new vasculature. That is the new vascularization. That is the angiogenic pathway. So whenever a body or a cell is encountering hypoxia, it will start growing the new vasculatures. And this is also a main problem in the cancer. You know, the tumor growth. What is the tumor growth? That is the angiogenic signal is upregulated. And when the angiogenic signal is upregulated, because that tumor, that particular tumor requires a lot of nutrients and oxygen, which can be done by the new vessels formation. So, HIF1 alpha, VEGF, and HSP90, this comes under the angiogenic signaling pathway. Apart from the MAP kinase pathway, June P38 MAP kinase pathway, there are other pathways which comes under the angiogenic signaling. So what is happening in the angiogenic hypoxia? The moment you restrict this HIF1 alpha and VEGF gene upregulation, cells will survive. But if it's downregulated, then there is no chance of cell survival. Likewise, what is happening here? That okay, if there is a misfolded protein caused due to hypoxia, it can be cured by the ubiquitin E3 ligase. I will discuss about the chip and parkin later on, that how chip and parkin acts as a molecular chaperone as well as an ubiquitin E3 ligase. And suppose if we cannot control this, if the pro-apoptotic proteins will be activated, which is the apoptosis pathway, you heard about the caspase, then caspase will activate and it can cause the apoptosis. But in this cartoon, we can control either by molecular chaperone or by ubiquitin E3 ligase. I will restrict my talk in these two proteins, because these two proteins are working as ubiquitin E3 ligase. And as I told you that molecular chaperone role is to give a proper function of a non-functional protein, but beyond a threshold, it has to discard it. So these two proteins can do either protection or destruction. And that was the reason why we have decided to work on these two proteins.
So, one clarification is needed. Most of these neurodegenerative diseases are not because of the misfolded proteins, it is the starting of the cause, but it's mostly the aggregation that is actually creating the problem, right?
Yes.
So, when you're talking about the misfolded proteins and this particular chip and protein that is you are talking in terms of basically the aggregation of it.
Yeah, what is happening when there is a misfolded protein, there is the internal dynamics, molecular dynamics of that protein. So what is happening? It's just a start aggregating because of the secondary structure is building. And this is the reason why you can have the aggregation of the misfolded protein.
Not all misfolded protein will give us aggregation.
Yes, you are right.
Because for example, your Alzheimer's or any of your Parkinson's, the age actually plays a role. If you say that. Even only at a very old age, you find that all this aggregation takes place. At a very young age, you still see the misfolded proteins, but you don't find any.
Because, okay, please continue.
Yeah.
Because what is happening in the younger age, our system, our machinery is very efficient.
Sorry.
But the moment we are getting aged, and this is the reason you can, and you have heard about a disease you might have, this frontotemporal dementia. You won't believe that frontotemporal dementia can occur at the age of 40. While the AD is occurring in the aged person. But there is a lot, if environmental toxins, if we are continuously encountering environmental toxins, it may happen in the earlier age also. Because when I was in Boston, I was working on this disease with the human samples. And you won't believe that we have worked with the 55, 56 years old AD patients also. That's biopsy sample. Sorry, autopsy sample. So it's depending. In Korea, one drug exposed and about 22 students were induced with the Parkinson's disease at the age of 19 years. And this is the reason how we are making the models. In the rodent model, just 1 year old model we are developing. So something can happen, but yeah, in normal way, it is always happening in the aging person.
In other words, it etiology of the disease you will not know actually still.
Etiology is, it's a various mechanisms, various hypotheses, but not at all convincing. Because, suppose a person is suffering from Parkinson's disease, there is only worldwide drug called levodopa. If a person is suffering from this Alzheimer's disease, only one drug which is called the memantine. Yeah, you can make a lot of drug out of that, but the real UFDA targeted drug is only memantine. Like in ALS, monoclonal. So these kind of things, means a lot of people are working. But I must not say like that, but for the sake of papers, there is no clear-cut convincing result so far we have got.
So what are the tests by which this FTD and AD are actually diagnosed?
Yeah.
Because in general, symptomatically they may appear to
Yes, like suppose if one day you ask me, hey, do you know me? I said yes. What is my name? So these kind of alarming situation, that immediately you can go, oh my goodness. Suppose you can give me a check without signing, and you will pretend or you will force me that no, I have already signed that check.
No, what I mean is by medically, how do you figure
Medically,
out whether
yeah, medically by
because
the
if you're saying A beta is responsible, so that means are there some tests by which you can validate whether the person is
Molecular
really
markers.
has AD?
So it's a Pittsburgh, this one molecular markers are there.
The problem with the AD and PD or NG is we cannot take out any kind of biopsy sample from here. So what we have to do, we have to take only the blood sample. In the blood sample, the problem is the blood sample. At the time when we can take the blood sample, there will be nothing appear. Only it will appear when it's a very severe condition. And when we can identify the severe condition, what is the use of that?
Yeah, that is why there's so much of research on early diagnosis. But the challenge with the early diagnosis
It's
is you
very,
need longitudinal data.
Exactly. Because how we can, okay, in an autopsy sample you can do any n number of experiments. But initially you can have either serum. Okay, come to the cerebrospinal fluid. It's very painful to draw the cerebrospinal fluid from the lumbar region. It's just like, means, it's like...
Aren't there PET studies by which they're able to see the A beta?
No, it's very difficult to understand. You can, in the PET study, you can just see the normal brain structure. But A beta is a neuronal structure is very hard to recognize. Because, sometimes this axonal bundle, sometimes this dendrites appears like, it's plaque. But because the plaque structure is also like fibular structure. So it is very difficult to diagnose in the early phase. And you can only diagnose in the mid or severe phase. And when you diagnose this mid and severe,
At that time, symptomatically
Yes.
also the subject is actually evident.
so this is the reason why a lot of people are working but no successful achievement, I would say like that. Okay, so what happened? We did some screening of medicinal plants. That okay, what we can use, what we cannot. So these are like some different polyphenolic flavonoids, and we use these biomolecules for the to check the efficacy of these biomolecules on the hypoxia animal model as well as PD model. So what we have done in the next slide. So here we have generated the hypoxic animal model. So it's a 8% oxygen here, and we have, this is a desiccator. We first precondition the animal, and after preconditioning, it's about 1 month preconditioning. And what we have done, not from 21% oxygen to 8% oxygen, rather it's a sequential, 21, 20, like that, and we narrow down to the 8%. When these models, when these mice are started behaving, means like comfortably in the low oxygen tension, then we have started this experiment for another 20 days, and after that we have started giving the drugs. Likewise, in the routine on what we have done, we have taken the rat, and after this rat, what we have done, we have just introduced this rotinone, 4 mg per kg body weight, and we generated the model. And how we have checked this model, that okay, what is the behavioral impairment? There are different kind of tests in the neurological disorder, like, suppose if a person is suffering from Parkinson's disease, if the doctor, if that patient will go to the doctor, what doctor will do? Okay, you put your thumb in my palm, and withdraw this. So for normal person, it is very difficult. It's very easy to withdraw. Okay, but for the patient, it's nothing. They cannot hold this thumb, and this is called the ball and palsy movement. In the ball and palsy movement, what is happening? If your thumb will be, or if your patient thumb will be held by doctor, it cannot. It's like he or she cannot hold. So what we have done here, we have generated the hypoxic model as well as the rotenone model of the PD. Rotenone is an environmental toxin and it impairs mitochondrial complex 4, so that the cytochrome C release and apoptotic pathway is triggered, and thus cause the neurodegeneration. However, it's a debatable topic right now, that whether rotenone is directly associated with the apoptotic pathway or some other things are also involved. It's require a lot of different investigations. And now this is your topic. I just brought 4 slides so that you can have some kind of feeling that okay, this is the computational biology touch. So we have done this Lipinski filter rule. We have used these drugs to check whether these drugs are feasible to use for the AD and PD. So this is the docking sites where can it work? Like for the DJ1 and Parkin. I will explain you why DJ1 and Parkin. In the Parkinson's disease, there are 5 different genes: alpha-synuclein, Parkin, DJ1, PINK, and LARC. So if all the 5 proteins are working perfectly fine, body is happy, brain is happy. But any mutation or loss of function of any of these 5 genes occur, the body is stuck behaving badly. And this is the reason why we have taken DJ1 and Parkin. Again, I can tell you, DJ1 and Parkin has also both the activity, chaperonic activity as well as ubiquitin E3 ligase activity. And this is the reason we have taken 3 different molecular chaperones as well as ubiquitin, CHIP, Parkin, DJ1. And not today, but we have also successfully identified that these 3 proteins are also making a very strong complex which can attenuate the toxic metabolites caused by the body. So straight away come to my topic. Then okay, biomolecules mediated neuroprotection. So what happened? As I said you that we have generated this model. So how to test? So after successfully duration of the experiment, we sacrificed the mice and dissected out the brain, and we took the brain slices. And on brain slices, first of all, we have done the silver staining. Silver staining is a very easy and normal technique to check neurodegeneration. And how it occurs? Whenever the neurodegenerated part, the silver molecule is precipitated in that part, and if you can see under the microscope, you will find it blue, big black. So you can see it here that we have done 3 types of hypoxic experiment. Normoxic means 21% oxygen. Acute, when the mice is trained and preconditioned with the 8% oxygen only 1 day, and that is acute. While the chronic means not for 1 day, rather 10 days of the 8% oxygen tension. So normoxic, which means 21% oxygen. Acute, 1 day 8% oxygen after preconditioning. Chronic, 10 days after preconditioning of 8%. And you can see it here that when we did this silver staining technique, we have found that in the cortex region and the hippocampus region, we have seen the dead neuronal cells. So this is a very strong technique. In the next figure, I will show you even a better picture of the neurons, how it looks like. So here we have seen that under chronic situation, we have got the heavy damage of the neurons. You can see few here in the acute, but not as good as here in the chronic conditions.
It's for Alzheimer's or what?
No, it's a hypoxic model. Hypoxic mediated neurodegeneration. Okay?
Because I was very surprised, basically striatum is not affected because Parkinson striatum is used.
It's a hypoxic model. Okay? And here what happened? We have used this naringenin, quercetin, and we have observed that after treatment of these two drugs, we have somehow rescued the neurodegeneration which is occurred due to the hypoxia. Likewise, we have done, now here is the striatum, what you are looking for. And here you can see in the striatum and substantive nigra, which is the most important part for the TH, means tyrosine hydroxylase production. Here you can see in the rotenone, there is a heavy load of this deposition of the silver particle, which is reflecting the neurodegeneration. But the moment we add the cis-mol and naringenin, it is rescued up to a very great extent. So here we have thought, okay, it's good that we have neurons, we have the PD generated neurons, but it can be reversed with the help of different drugs like cis-mol and naringenin. Okay, so now we checked if there is a neurodegeneration, there must be some neuronal dysfunction also, or this morphology. Let's check the morphology, and what we have done the TH staining. TH is the tyrosine hydroxylase enzyme, which is giving you the dopamine in your brain. Okay? So suppose if there is less TH, less dopamine production. More TH, normal level of the dopamine. So here, if you can see the striatum and substantia nigra, under the cis-mol regime, neuron is very healthy, happy, no issue. So what we have done the Western blotting. I hope you know about the Western blotting. It's the SDS page, sodium dodecyl sulfate. It means we are checking the protein at the molecular masses. So when we did this, what is happening here? You can see that in normal, it's not a very good blot. I'm sorry for that. But rotenone, this is the naringenin, this is down-regulating. The TH is more or less equal to normal condition. In the rotenone, TH is high. Actually, this is not very distinctive here. So, but here, in the before slide, you can easily see that in cis-mol, the morphology of neurons is very happy and healthy. Okay. Now, in the upper panel, it's a hypoxia-induced neurodegeneration. In the lower panel, it's a rotenone-induced PD model. Hypoxic model, PD model, Parkinson's disease model. And when we check the level of different heats of proteins, we found, and heats of proteins are protective proteins. So wherever there is a retinol, the level of HSPs are down. But when we use the biomolecules, the level of heats of proteins have been rescued. And you can see it here that level of HSP 90 in the chronic condition, but here these are chip, Parkin, HSP 70, HSP 90. Likewise, in the same kind of experiment we have performed, and you can see in the rotenone, what is the level of HSP 70, but what is the level of HSP 70 after cis-mol treatment as well as the naringenin treatment. So these kind of experiment we have performed to show that indeed these biomolecules can rescue or at least little bit upregulate the level of different protective proteins such as mix of protein HSP 60, 70, 90, chip and Parkin in both the models.
So you know the mechanism of action.
Mechanism of action. Okay.
These biomolecules.
The logical extension is basically what is the molecular mechanism responsible for upregulation of HSP, because you also mentioned about DOPA, with basically the diuretic property, pyroxene. How does HSP 90 and pyroxene hydroxylase play? Because pyroxene hydroxylase is a autocatalytic reaction, which actually increases your DOPA. It actually transfers your DOPA from outside to inside. So, what is the role of HSP 90 coming into the picture in this metabolic?
Okay. So let's talk about the transcription and translation. Okay. Whatever induction we are giving to the cells are trying to transmit that kind of protein. Let's say how cells will be associated. We have done the IP experiment also, immunoprecipitation experiment, and we have successfully established the fact that indeed these proteins are interacting with TH as well as the beta, as well as the Parkin gene, as well as the different genes. So we have done this IP experiment in order to prove the mechanism of action which is associating in the complex. Yeah.
So does that mean that genomic analysis can be used for early diagnosis of the, or is it being done?
Again, problem is we have only sample like serum and blood, and it is not means like seen that a lot of A beta deposited in the blood. It's not circulating in the blood.
Then what I mean is that if there is any mutation because of which protein malfunctioning is happening or something is happening, maybe it is detected in the at the gene
You
level.
are absolutely right, but at least the level of that particular gene should be upregulated. Then only we can detect.
That, so that doesn't. So that is why. So it's that in genomic analysis is not being used for enzyme.
Again, you can use the genomic analysis, but in the later stage.
Latest.
Here we have a collaborator in the Gangaram hospital. We have 20 PD patients' blood, but in the 20 PD patient blood, you can't find anything. Nothing. It's simply you can be very happy that okay, I have 20 blood samples from the PD patient, but that blood is useless for PD researchers. And it can only be expressed when the person is completely disabled. At that time, if you can, it's useless because you know by the
clinician will just by looking
by
at
looking.
the person.
Wall pulsing movement, okay. Yeah, please.
Is there any scope left, or is that you could not detect any known markers? Or is there any scope in the blood? Because there could be some cell-free DNA. In cancer, liver cancer or something.
There is a big difference between the cancer and the neurodisorders. Okay, because with neurodegenerative disorders, one big problem also, whatever drug you are giving, it has to cross the 3B, blood-brain barrier.
Yes.
Okay, but in the cancerous situation, many genes are upregulated, and you can just have some blood, and that's it.
That's what I was saying.
But here, the high level of antigen can only be occurred in the severe condition, not in the early condition. Now, one may ask, then why the hell neuroscientists are doing work? We are just with this hope that maybe someday we can find something. But problem with the human patient, we cannot take in the animal. There is a lot of established fact, but in animal we can sacrifice, but in human we next to impossible thing. Yes, next. Because suppose if a person is working, can they give a little bit part of the brain? Okay, sir, you have a problem in the hippocampus. Can we have it from here? And what would happen? So this is the problem. Even in the when I was in Harvard, at that time, you know, Harvard brain research resources is very big. You just order it, fax it within, not like in our own country that we keep on just telling you so many things, but not give you a single sample. Over there you just, okay, BRAC5 area, 5 sample, temporal lobe, 5 sample, and 5 control. 15 samples, just call in the morning, and by 12 o'clock you will have that sample. Do research whatever you want to do. Here in NIMHANS, I have tried my level best to get a single sample, what I got the answer. After 1983, no 80% sample is here. So we have only one AD sample in India. In the 1.25 billion population, there is only one AD sample. And they are calling themselves that they have the brain bank. So no, this is the hypocrisy. I agree.
So they have, they don't want to give, or they don't?
Yeah, they don't want to. They have invited my CV 5 times. And I don't know what they have done with my CV, but anyway. Let us not discuss on this thing. It will just So, come to the restoration of the protective protein upon sysmol. And now, in one shot, I am trying to show you the Parkin chip and DJ level under normal condition, under rotenone, sysmol, and naringeline. And you can see that in the, just concentrate on this panel, that under rotenone what is happening here, but the moment you add the sysmol, these protein levels are upregulated with respect to the rotenone, and come to the normal level here. So we have successfully shown in the 3D model that indeed Sysmol and Naranjini can rescue the protein level of Parkin, Chick and GK1. Yeah, go ahead.
Rotinol is the neurodegenerative inducing factor. Sysmol and marginine are the flavonoid compounds that which actually put back reverses your Now, also another question is that like you also gave Sysmol and marginine along with the rotinol, right?
No. First of all, we have generated the model. When that model is completely
okay. So you gave a rotinol.
Yes.
Then afterwards,
Yes.
induced
like Madam asked that okay. First, we have checked that okay means behavioral effect, and then we have started giving the.
Okay, so.
Actually, no. This Small is now widely using. We are the first group who has reported the entire study because so far Small has a role, medicinal role, but nobody, no group has shown this extensively the role of rotinol. So yesterday was my professor interview in my same department, and they were asking the same question that okay, how have you used this Small? But it's not a big deal in that because I was not giving anything orally. It's like a sesame oil. You just rub, just take it, just lukewarm sesame oil, and just rub in one direction from one side to either lower to upper or upper to lower in both the muscular thing. And we have seen many patients as a better effect, not that like completely cured, but at least they have a better effect. In the PGI Chandigarh, they were heavily using this in England, in US. These works have been praised heavily, and they are using this oil only. They are not giving anything orally. They are just using massaging with this oil, and the effect is good. But again, the effect is in the early stage, not in the later. Later, nothing is working. Go ahead. After a long time, I am having some. Because most of the time, whenever I am speaking, I am only speaking. Nobody is asking. Yeah, please.
So, one of the things is that how do you come to know that this flavonoid compounds are going to be the active generative one of this particular I mean,
Super question.
How do you know that, like, how do you actually come to know about it?
Yeah, I can tell you. First of all, most of the neurodegenerative disorders are also occurring due to oxidative stress. And flavonoids are the good quencher.
So it's a medicinal
It's just a hit and trial method. Okay, I have found that few like Small, and when I was thinking that why Small is so it's a very small compound, C7H6. Sorry, C7H6 and O3, like very small compound. So we thought that why not we can use it? It's a phenolic compound, why not we can use it? And I can tell you one more thing because I am always encouraging students at least and researcher colleague that suppose if any of us will read the Charak Samhita, you won't believe how many patents we can do. Although I am against the patent, but how many patents we can file, no one can even think of it.
But if it is, okay, I think patenting is not the point, but there is interesting knowledge out there.
Charak
Because if it is already
Charak
Charak
Samhita,
Samhita, probably we won't be able to do it.
Yeah, in my name there is no patent. Although I can entitle for 8-9 patents, but I have no patent because I am not in favor. But if somebody will read Charak Samhita and understand, they can find millions of molecules, biomolecules, which is available in our country.
So flavonoids and coumarins are the two
Yes,
very important
curcumin, yeah.
But same time, when you feel about the small molecule, then the toxicity is more because it can go anywhere.
Yes, sir.
That's a major part of the problem.
Yes,
with the
yes, sir. You are right.
most promising molecule which shows in the experimental studies that this is working, it means there is a lot of toxicity. We have to be very careful about it. Even the Lipinski route is not a good way actually.
Sir, I agree about the Lipinski rule. What I am trying to tell you, sir, that we have to check the LD50 value of particular drug first. If I am working on the wet level. And second, what is the bioavailability? If these two things are working properly, we can do this kind of experiments. Otherwise, as you said, curcumin. Yes, we are eating a lot curcumin. So theoretically, we must not have anything like cancer, like any neuroinflammation or anything. But
Everything we have.
You tell me, means like in every house in India at least, curcumin is used heavily, means haldi. But what is happening? So you are right, sir. The bioavailability and Lipinski rule is just like to satisfy yourself that okay, you are doing something good. Otherwise, I don't. Sorry,
Because
sir.
body is too complex. That how much toxicity will be there, it will not be clear in 1 or 2 studies. It takes a long time.
Yes.
So sometimes the drug you are giving today will be harmful in the long term.
Yes.
So very difficult to
Why
understand.
this study is successful? Because we have sacrificed the animal after 30 days. And in the computational biology, please don't take it otherwise, you can tell me that okay, this drug is active. But if somebody will ask you, okay, prove it, what you will do?
But with the computational biology, we have to understand very clearly. It helps you to prioritize the drugs.
Yes, you are right.
Otherwise,
That's what we
randomly
have
how
done.
many you will try?
Yeah, that's what we have done.
So-
because we have to narrow down our experiment. We cannot use 500 molecules for animal or for in vitro condition.
So it is important to understand the limitations as well as the advantage of any technique.
Exactly.
So computational biology, biotech, this is a technique.
Exactly, sir.
It can
Like in
help.
genetic engineering, if somebody is saying that okay, I am a specialist, genetic engineering is just a mere tool.
Yes.
Now you have to express the protein, otherwise your clone is useless. Like here, as sir has pointed out, obviously you should have means like limitations. You cannot go for the 50 biomolecules. Even 4 or 5 biomolecules is more than sufficient to carry out 5 years research. So that's why we have narrowed down our experiment with the help of bioinformatic tools. Now, as I said, use this rotinone, which is if following the apoptotic pathway, what would happen in the caspases level? And here, in the upper panel is the hypoxia mediated neurodegeneration, and here is the rotinone. And you can see here that in case of chronic hypoxia, in case of rotinone treatment, we have found the cleaved caspase level, caspase 3, which is the executioner, and caspase 9, which is the initiative caspase. But the moment we treated with these two models with the cis-small naringenin and quercetin and naringenin, we have found no cleaved caspase level, which indicate yes, apoptotic pathways are involved, initiation of caspases are involved, execution of caspases are involved, but the moment we have added these two drugs, no more cleaved caspase level, no more degeneration, no more apoptosis triggering. Finally, we did another experiment of the ubiquitination assay. So suppose in the case of acute and chronic condition, we have got the polyubiquitination band. This is the polyubiquitin band in both the condition. But the moment we added this, the ubiquitin level is dropped down, and the best ubiquitin drop down is in the case of chronic and naringenin treatment, which means that ubiquitination is also inhibited after using of the naringenin and quercetin in the hypoxic mice model. I don't think that it's a combinatorial approach of this drug, so it's a, okay. Now come to this slide where we wanted to show, okay, if the PD is a neuromuscular disorder, what is the effect of rotinone on muscle? And here you can see that this is the rotinone plus cis-small, this muscle fiber is appeared here. This is the control versus the cis-small, but in the rotinone, it's a sort of atrophy. So what we have done, the same animal, from where we have dissected out the brain, we have dissected out the muscle and stained with the and stained this muscle, and we have found that indeed there is a rescue of the muscle morphology. This is the different heats of protein and ubiquitin 3 ligase paradigm in the muscle. This is muscle blot, and we have again shown that in case of cis-small and naringenin treatment, the different protective proteins are upregulated in contrast with the rotinone alone. Then we have done this polarization assay of the ubiquitin and caspase, because if apoptotic pathways are triggered whether it's following the ubiquitin pathway or not. So we have checked this and we have successfully done this, and you can see that ubiquitin cascades after merging. In case of proteasome, you can see the green and red is yellow, and yes, these are the co-localized in the same tissue muscle fiber, while upon adding of this small, this is gone. So we have checked the brain, we have checked the muscle, we have checked 4 different drugs, and we have proved that indeed these pathways are active, and somehow we can rescue with the help of the small and very engineering. One interesting fact I just want to tell you regarding this crosstalk between AD and PD. Here, this is my work which I have done in the when I was in Tufts. So this is the Alzheimer's disease brain sample, human brain sample, and you can see this is the IP experiment what we have done. So in the control, there is an indulgence level of A beta, which is the monomeric form. But in the AD patient, you can see this is the heavily expressed upon blotting with the A beta antibody, and this is the peptide is the control. This is the inclusion body myositis, and you can again see here in the transgenic animal of the inclusion body myositis, we have found a considerable level of the A beta and oligomeric pattern. While in the PD also we have found the A beta. But now here is the interesting fact. There is a crosstalk between the Parkin and A beta. If an animal is expressing more level of Parkin gene, there will be less level of A beta. And if that animal is expressing less level of Parkin, more level of A beta, which you can see it here, that this is the wild type and knockout. In knockout, this is the Parkin knockout model. Here, no Parkin, more A beta. Here, you can see more Parkin, no beta, no A beta. You understand this lot? No Parkin is here, lot A beta. More Parkin here, no A beta. So we have successfully did this experiment, and there are many other experiments which we have done to prove this point with several IPs and several immunohistochemistry experiments to prove this fact in the animal model as well as in the human autopsy samples.
So
Yes,
that
please.
means that a person can have Alzheimer and Parkinson at the same time?
No. If a person is suffering from Alzheimer's disease, he is not going to have the Parkinson's disease, because in the Alzheimer's disease, A beta is upregulated, Parkin is downregulated. So this is my previous team when I was in VIT University, and this is my current team. And these are my academic assignments. I am the honorary member of the E-Pathshala also. I don't know, it's a UGC MHRD program. And these are my ongoing projects in the lab, and nucleophilic ligase and chaponic pathway in the involvement of neurodegeneration. I have started this in silico study because I don't have much knowledge, or you can say I have no knowledge in the bioinformatics. I have only used few while doing the cloning and all this. Otherwise, I don't have much idea about the bioinformatics. Cell cycle dysregulation in PD is also a very important field for our research. Identification and characterization of drug molecules of the neurodegeneration. Obviously, I'm planning to do more work on the AD-PD crosstalk. And recently we have published a paper in Journal of Alzheimer's disease where we have given some glimpse of the ID neutralizing in the AD pathology, but lot has to be done. Hypoxic induced neuromuscular degeneration in the mice model, and again I just wanted to continue with this work if possible here, and these are my publications. Thanks for your attention. Yeah.
What could be the pathway? Is it not DJ beta?
Yes.
Is it what?
Not also.
It's not.
Yes.
Notch is already
Previously
one
I
of
have
the
found.
secretest pathway.
Yes.
So notch should be actually clear
Yeah.
the secretest.
Previously, I actually found March being distributed in Alzheimer's. What is something to not and Parkinson and
Yeah, we can do it together. You just give me the brief idea using the bioinformatics tool. I will do the wet lab experiment.
Yeah, most definitely.
So, coming to the softwares that you use for the bioinformatics studies that you have done, so which kind of tools did you use? And the protein structure is available, right?
Yes,
The one which you use. And for the docking kind of thing, what kind of software did you
This
use?
is one autodoc program.
The free software.
Free software, yeah, we don't have a
So we are checking so that even our students are reacquainted with that. So we are using Autodoc for doing the finding the molecular mechanism, or is it for the screening purpose?
only screening purpose.
Screening purpose. And how many molecules did you screen?
We have screened about 7-8 molecules out of it's like a lot of words, it's like 200 or 100. In that we have found based on the Lipinski filter rule, but as Sir said, and I am also
That's right. It's
it.
not often always valid.
Because this small things I have read somewhere that it is also neuroprotective. But apart from that, nothing has written. So I thought that it's antioxidant, it's a neuroprotective. Let's start with this experiment. And this small is तिल. हम लोग तिल का लाई खाते हैं ना, वो मकर संक्रांति में सोचे कि चलो एक बार ट्राई किया जाए. और अच्छा ही आया है रिजल्ट.
So is it neuroprotective or neurodegeneration? Regenerative also.
You can say both.
Like Prozac, it's called as neurodegeneration.
But you can say both. Because if there will be neuroprotection, then only regeneration. And if there is a neurodegeneration, there is a neuroprotection. What? No. For that we have to work little on stem cell. But stem cell again, I don't think that how, because these days in India, for the PD patient, stem cells are frequently used. In Bangalore, in Hyderabad, these two places I know very well because one of my relatives is suffering from PD, but they have been rescued with this stem cell therapy. Although they have also tried my things, but initially it was okay, and after that.
So you have mouse and rat model for PTSD?
No, I have mouse model for hypoxia. I have a rat model for PD. Because if you can induce the rotinol in the mice is very 50 gram to 100 gram, but rat up to 300 gram or 400 grams. So it requires body robustness also to cope up that medicine. Yeah, please.
So, what's the role of genes? We always talk about only the neurons, because nowadays, why I specifically ask this question is that after 50 years still we are actually holding on to amyloid. Amyloid proteins, that might be the biomarker or something, but as of now still, after studying the amyloid for almost 50 years, still we are unable to find a cure for that. Right? So, the reason being we are looking only at the folded proteins and this thing we are looking at. But we are not taking into consideration of the glial cells which are present.
No, glial cells we have also done experiment. Not me, but many groups have performed this experiment. But again the problem, because glial cells is still dividing in our brain, all with very little numbers, but it still is dividing. Only glial, egg and sperm, these three are dividing. But the problem with these glial cells, what we can do with the glial cells?
There must be actually
No, I'm just trying. If our hippocampal region is gone, what is trying to prepare the neuronal cells? But what with astrocytes or basically but the problem with that if the most unfortunate with the neuroscientists are that the moment we know, or the moment we knew that okay, these things have happened, time is gone already.
So that's another reason that, for example, a familial, for example, two generations of the people have got Alzheimer's or something like that. The third generation, they don't want to go for the test.
No, I can tell you, initially it was the hypothesis that PD is due to genetic factors, but now it's obsolete. PD is not,
It's not a familial.
No.
So that's one thing. Second, one final question might be.
Go ahead.
Can you just throw some light on what is the cell division cycle you are talking about in terms of starting?
Okay. What is happening that post-mitotic cell division is always lethal. And we have published so many papers recently that post-mitotic cell should not be occurred inside our body because it's lethal. And how it's lethal? because it's activating different cyclins, which are the cell cycle markers. So what happens, suppose if you follow the rules of the traffic, everything is fine. If you are not following, things will happen bad. And here again the same thing, that if a cell is jumping the checkpoints without permission, this is called the aberrant recycle entry, and this is the post-mitosis
That
element.
also happens in cancer, right?
Yes,
Complete
yes,
breach of
yes,
the
yes, yes, yes. Hyperplasia and hypertrophy, yes. Abnormal cell proliferation.
Okay, so how does this cell proliferation differ from
Okay.
what it's
The moment this cell cyclins levels are upregulated, it follows the apoptotic pathway, and we have successfully did this experiment, and we successfully published all the human molecular genetics this in 2014. Not only me, hundreds of groups are working on the cell cycle level. What is happening in the PD patient? If you can check the brain slice of the PD autopsy samples, you will see that an increased level of different cyclin genes in the PD patient, and as well as in the AD patient.
Is there any specific phase that which is actually breached, or it's all the phases, all the checkpoints at G1 is breached?
No, basically it's a G1 to S initial.
I think Notch is involved in one.
Notch is involved in AD,
But
but
that's a signal transmission.
Yeah.
Okay, you're talking about
signal transaction you can correlate any possibility.
Yeah, it is. Because like if you take a hash graph and map your object.
Because here I have not shown you one thing. In the AD patient, in the same patient here, we have seen cyclin D1 upregulation, cyclin E upregulation, which is S phase marker, and cyclin A upregulation.
So that's all G1 to AS.
Yes.
Alright, so let us thank Professor Pravir Kumar. Please. So this is an institute.
Thank you, guys. I heard a lot about you, sir, and I have seen Dr. Yasa, I think. Oh, Ravi, Arkeshna, sir. So, we left that place, Chandigarh. Yeah, we are fortunate. We are here.
Okay, so we have refreshments and the speaker will also be available to you to ask any questions that you may have.
So I think